Every living cell carries a universe within it, millions of molecules talking, reacting, and deciding our fate, and there are thousands of layers of genetic regulation. But among them exists a tiny messenger, smaller than 24 nucleotides, yet powerful enough to shape life and disease. This is a story of microRNA or miRNA. Though only 20 to 24 nucleotides long, miRNAs have become recognized as one of the most influential controllers of gene expression in modern biology. What these miRNAs do: microRNAs don't build proteins themselves. Instead, they decide which proteins are allowed to be made. They bind to messenger RNA, the blueprint of the protein, and silence the message. Sometimes they destroy the mRNA completely, erasing the message before the cell can even read it. Other times, they simply block translation, holding back the ribosome and stopping protein production. Through this mechanism, miRNAs fine-tune how much a particular protein a cell produces. In silence, they control expression. With this quiet control, miRNAs orchestrate some of the most fundamental events of life. They guide cells as they grow and divide. They drive tissue development and differentiation. They decide when a cell should live and when it must die, or you can say programmed cell death. They fine-tune immunity and metabolism. Without miRNAs, the fragile balance of life would collapse. To carry out their regulatory functions, miRNAs go through a multi-step maturation process. Inside the nucleus, a microRNA gene is first transcribed by RNA polymerase II, producing a long primary transcript called miRNA. This transcript is marked by METTL3 with an M6A modification which supports proper processing. Next, the drosha DGC-R8 complex cuts the miRNA into a shorter hairpin-shaped molecule known as pre-miRNA. Exportin 5 along with RAN-GTP then recognizes this precursor and transports it from the nucleus into the cytoplasm. In the cytoplasm, Dicer together with TRBP further processes the pre-miRNA, creating a double-stranded miRNA duplex. One of the two strands becomes the mature miRNA, while the other strand is removed. The mature miRNA is then loaded into the RISC complex, which guides it to its target mRNA. Based on how closely it pairs with the target, the miRNA can either block protein synthesis or trigger degradation of the miRNA. In both cases, the final outcome is reduced protein production. Additionally, in certain contexts, some miRNAs can activate immune signaling by binding to toll-like receptors, which plays a role in inflammation and cancer. A single miRNA overexpressed or silenced can trigger devastating consequences: cancer, neurological disorders, cardiovascular disease, diabetes, even viral infections. All carry the fingerprints of miRNA dysregulation. Overexpression or loss of specific miRNAs can alter key signaling pathways contributing to these diseases. Today, miRNAs are considered critical molecular indicators of health and disease. And that opened a new chapter. If miRNAs contribute to disease, could they also heal it? Scientists created miRNA mimics, synthetic versions designed to restore beneficial miRNAs that were lost. Delivered through nanoparticles, viral vectors, or even exosomes, these mimics reactivate natural gene silencing, teaching diseased cells how to protect themselves again. By re-establishing natural gene regulation, miRNA mimics show promise for treating conditions where protective miRNAs have been silenced. But sometimes miRNAs become villains, too powerful, too destructive. Some diseases are driven by excess miRNA activity. For these cases, researchers developed miRNA inhibitors, antago-MIRs and anti-MIRs, molecules that block harmful miRNAs from binding to their targets. Like shields, they defend essential proteins and restore cellular balance. These inhibitors aim to prevent abnormal suppression of essential genes and restore cellular balance. Beyond treatment, miRNAs are transforming diagnostics, and perhaps the most extraordinary role of miRNAs lies in what they reveal. Because they are highly stable and they circulate through blood, serum, urine, saliva, carrying clues about disease. Long before symptoms appear, miRNAs serve as powerful non-invasive biomarkers. As biomarkers, they offer hope for non-invasive diagnosis, prognosis, and treatment monitoring, a window into disease hidden in plain sight. They can provide early detection, help evaluate disease progression, and guide personalized treatment strategies across a wide range of disorders. So, in the end, miRNAs are more than tiny strands of RNA. They are guardians of genetic expression, gatekeepers of health, and messengers of disease. And one day, miRNAs may not only explain illness, they become cure. From gene regulation to disease diagnostics and emerging therapies, miRNAs have reshaped our understanding of molecular biology. As research advances, these small RNA molecules continue to reveal new possibilities, not only to study disease but to predict it, prevent it, and ultimately treat it.